In this video, we will be discussing our second macromolecule group called lipids. Lipids are foods that such as butter, any type of oil like olive oil or peanut oil or vegetable oil. We also have animal fat that comes from certain meats, grease, anything like that would be considered a lipid. They are large nonpolar organic molecules. Now, nonpolar is a very important term when it comes to lipids. Nonpolar means that it does not dissolve in water because water is a polar molecule. And what it means to be polar is that the molecule itself, when it's stable, has a slightly positive charge on one side and a slightly negative charge on the other. So in the water molecule of H2O, the oxygen is a slightly positive portion of the molecule, and the hydrogens are slightly negative. Now, lipids, being nonpolar, they do not have any charge whatsoever. They're completely even. There is no slight positive or slight negative, which means that anything that is nonpolar and it comes in contact with water will not mix at all. The structure of lipids make them repel water, as I stated before. If you've ever tried to mix water and oil together, or vinegar and oil, anything, with oil that's water soluble, it's never going to mix. Like oil and vinegar, if you were to shake it, you may get, certain bubbles of oil that, start to mix in with the water, but eventually it'll settle out. The elements that make up lipids are carbon, hydrogen, and oxygen, which are the same elements that make up carbohydrates. The difference in structure between carbohydrates and lipids is that even though they have the same elements that make them up, they do not have that ratio of 1 to 2 to 1 like carbohydrates do. A lot less oxygen molecules in lipids in comparison to the hydrogen and the carbon elements. As I said before, unlike carbohydrates where they have an even ratio of 1 to 2 to 1, there is no ratio. There are just a lot of hydrogens and a lot of carbons that form what we call these fatty acid chains, and there's a very small amount of oxygens which are just found in the tip of the lipid molecule. Most lipids are made up of carbon chains. As I stated before, these are called fatty acids, and these are the building blocks of lipids. So, in another word, these are the monomers of lipids. Fatty acids are the monomers of lipids. Fatty acids are the building blocks. They come together to form the polymer, which what we know are called lipids. If we take a look here of the model of fatty acids, all right, this is just palmitic acid. This is just one of the many fatty acids that there are. We look here at the structure. Now this is broken up into two pieces. Over here, we have everything I'm circling right now. All right, this is the hydrophobic portion of the fatty acid. Hydro means water. Phobic means fear of. So the word hydrophobic means fear of water, aka it does not mix with water. So this is the nonpolar section that we were talking about before. And what makes this hydrophobic is this wall that we have here of hydrogens. This hydrogen wall will separate and not allow the water to mix, because remember, water is polar, and these wall of hydrogens make this whole section nonpolar. Now over here, in this blue circle that we have over here, this is what we call a carboxyl group. Notice how we have oxygens in the carboxyl group. This makes this small section up here polar. So there is a really small portion of the fatty acid. It's only in this carboxyl group where you find the oxygen that is polar, and it can mix with water. So the fatty acid will arrange itself if it is near water in any type of environment, it'll arrange itself so this portion right here will be towards the water, this will be away from it because it repels. So we call this section over here hydrophobic, aka it does not mix with water. But over here, we call this hydrophilic. Hydro again meaning water. Phil means to like or to love. So we call this water loving. So the hydrophilic portion over here will mix with water. The hydrophobic portion over here will not. There are three major roles or functions of lipids. The first is that they store energy. So unlike the carbohydrates that we discussed before, where they have short-term energy, that was their function, lipids are going to store energy, what we call I'm going to type this here, long-term energy. I'm sorry, my handwriting is terrible on here. Okay, but that's important to know. So long-term energy is going to be the function, the major function of lipids, and like the carbohydrates where they are the short-term energy, these are going to be the long-term. The second role or function is that lipids are used as chemical messengers. So they're almost like these antenna-like structures that are found on certain proteins or on the outside of certain cells. And what they do is they allow cells to communicate with each other. For certain cases, like if you were to maybe get a scrape on your hand, okay? Eventually over time, those cells are going to divide and divide and divide until that scab or area where the cut was is now eventually covered up. But how did the cells know when to stop dividing, and how do they know where the cut starts and ends? That's where the chemical messengers come in. There are certain signals that one cell will send to another, and it'll say, okay, you've reached the end of the cut, you can stop dividing now. So that's another role of lipids. And the third and the major one is that lipids are the structural component of the cell membrane. So we've already discussed the cell membrane. We haven't gone into detail of what it actually is made up of, but there are these molecules that we call phospholipids. Phospholipids. They have two major portions to them. They have fatty acid tails, which, as we stated before, the fatty acids are the monomers of lipids. Those are the portion that are nonpolar and they are hydrophobic. They do not mix with water. But then they also have what we call a hydrophilic head, which is that carboxyl group that we discussed earlier. And there are two layers of the cell membrane, and the tails will arrange themselves so they touch near each other, which causes this entire zone of nonpolar and this entire zone of hydrophobic, where water can't get through. But on the very outside, where the heads are, and the very bottom on the inside where the heads are, water can mix just with that particular portion. And that's where, that's how the cell membrane is structured. The cell membrane is fluid, so these phospholipids are able to kind of move around, and that's what allows the cell membrane to allow certain things in and out. As we said before, it's a semi-permeable membrane, so there are, certain things that can go in, certain things that can go out, then there are some that just can't. It really just depends on, is it nonpolar? Is it large enough to physically fit through the phospholipids? There are many different reasons why certain things can pass through and why they can't. But it's the structure of these fatty acids and it's the structure of these lipids that make this hydrophobic and hydrophilic zone, and that's why the cell membrane is semi-permeable. There are 4 different groups of lipids that we'll be discussing. All of them are slightly different in their structure, but overall they're all made up of the fatty acid monomers. Therefore, they're all considered to be lipids. They also do not mix with water, which also gives them that characteristic of being a lipid. The first group is what we call triglycerides. Next, we have phospholipids. Third, we have waxes. And fourth, we have steroids. So let's get talking about triglycerides first. Triglycerides are made up of 3 fatty acid chains, 1 glycerol, and that's going to equal our overall lipid. So again, we're talking specifically triglycerides only. This is the structure that they have. So here you would say that we have over here, this is our glycerol head. All right, notice here we have our carboxyl groups. Carboxyl group here, carboxyl group here, carboxyl group here. This right here is our fatty acid chain. So this is one monomer. Here is another monomer. Here is another monomer. And this whole large chunk up here would be our 4th monomer. So we have 1, 2, 3, 4 monomers coming together to form this large molecule here, which we call our triglyceride. And if we're using a general term, we could just call it a lipid. All right, but this right here, this is a fatty acid, this is a fatty acid, this is a fatty acid. This is not a fatty acid, even though it's a monomer. We do have our glycerol. This over here is our polar portion, nonpolar, 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 which makes this partially polar and partially nonpolar. Now, if you notice here, through the dehydration synthesis process, which we've discussed before, a water molecule will be removed where each bond occurs. So we have water leaving, which allows this monomer here, monomer here, monomer here, monomer here. It allows them to join together to form what we call a triglyceride molecule. So our triglyceride here is our polymer, and we have 3 fatty acids, which are monomers, 1 glycerol, which is a monomer, coming together to form this one huge, large polymer called a triglyceride. There are 2 different subgroups of triglycerides. We have our saturated triglycerides and our unsaturated triglycerides. Saturated fats are solid at room temperature, and they only contain single bonds. So what that means, and if we take a look over here at our model, notice how there are one little dashes, right? There's one dash between each C and each H. Over here, we notice that we only have single dashes, which signifies that there are single bonds. If you look here at the bottom underneath, we have unsaturated fatty acids, and notice over here in these highlighted areas, we see 2 lines, and those 2 lines represent a double bond. So when you have a double bond, you are taking away the opportunity for there to be an extra hydrogen on the outside, because carbon can only make 4 bonds. So if one of them is taken away, that means that one hydrogen is not allowed to bond because one of them is taken away from the double bond in the middle. Now, when you think of the term saturated, you would think of almost like a saturated, something is saturated with water. It's completely filled with water, right? If you were to have a towel that was dry and you were to dunk it in, a pool of water or in a sink or something, and you lift it up and there'd be water dripping from it, and you would call that saturated. It means that it's hit its capacity for how much water it could hold. So here, we're not talking about water, but we're actually talking about these hydrogens over here. So because this molecule only has single bonds, we can saturate this fatty acid with the most amount of hydrogens they could possibly hold. And this is what makes saturated fats a little bit more unhealthy. Okay, so an example of a saturated fat would be butter. These are going to be coming from animal products, if you see up here. So we have butter is our, most common example of a saturated fat. And we all know that that's a little bit more unhealthy to consume versus our unsaturated, which would be something like oil, maybe any type of oil, olive oil, vegetable oil, peanut oil, sunflower oil, any type of oil, they are all considered to be unsaturated because they are all plant-based, and they also contain those double bonds. So if you notice here, there are gaps where there could be hydrogens, but there's not because the bond is occupied over here in this carbon chain by that double bond. So what that does is it causes bending because we have here these holes. So this molecule here is not as stiff, which causes it to be liquid at room temperature. But if you notice over here, the saturated fatty acids, there's absolutely no room to bend because it's completely saturated with these hydrogens. So therefore, it's a more stiff molecule, which allows them to be solid at room temperature. Next, we have our waxes. Waxes are very highly waterproof, or hydrophobic, as we said before, is waterproof, doesn't mix with water. We have examples here of waxes on a plant and waxes from animals. So let's start with the plant first. Plants have what we call a waxy cuticle. Only some of them do, not all of them. And that is made as a protective outer coating, so there's not too much water that's allowed to go in. It's going to protect the plant from too much water. Plants like this are typically found in areas where it rains a lot. So therefore, that waxy cuticle is going to allow this, the amount of water that the plant needs without being completely saturated by water. Over here, we have earwax. Okay, so this would be like the outer portion of the ear. In here, this is the earwax. It is 100% waterproof. And the function of earwax is to keep microorganisms from entering the inner ear. It also it blocks water from entering the inner ear, which would cause microorganisms to breed in there if it were to actually get in. But you don't want extra fluid coming from the outside going into your inner ear, so that is what the function of earwax would be. Steroids is our next group of lipids that we'll be talking about, and these don't actually contain fatty acids, but they do contain links of 4-ringed carbon structures that are hydrophobic. Their characteristics are very similar to a fatty acid, so that's why they are placed into the lipid category. They make up cholesterol, as you can see over here. Cholesterol is a hydrophobic, almost like this plaque type of substance that can get caught in your arteries. Cholesterol is a necessary molecule that we all do need, but in very low amounts. When cholesterol starts to get high, that plaque will build up in your veins, in your arteries, and it causes blockages, which is very unhealthy. They also make up the very most important hormones in our body, all the hormones, things like progesterone, and we have testosterone and estrogen. These are all hormones that we would not be able to live without. And steroids, they all fall into the category of steroids. Our last group is called phospholipids, and these are the molecules that make up the cell membrane like we discussed before. So we have here a picture of one single phospholipid. It's broken up into 2 groups. We have our hydrophilic head, and we have our hydrophobic tails. And we have 2 fatty acids that make up the tails and 1 glycerol head that forms the hydrophilic portion. So unlike the triglycerides, where we had 3 fatty acids, phospholipids are only made of 2 fatty acids and 1 glycerol head. They're very similar, but the only difference is that they have that 1 less fatty acid chain. Remember, this whole section over here, right, this is going to be nonpolar. So this is nonpolar. It's a terrible P. Nonpolar, and these are also going to be hydrophobic. Okay, and then up here, so put PO for phobic, and then we have up here, this is going to be hydrophilic, so we'll put, okay, PHY. This is so hard to write like this. PHO, so this is going to be our hydrophilic up here, and this is also going to be our polar, POLAR. Okay, and then over here, this is what I was saying before. So the cell membrane is made up of what we call a phospholipid bilayer, and the bi means two layers. So right here, this is going to be our top layer of phospholipids. This would be our bottom layer phospholipids. They are made up of the exact same thing. Remember I said before that the tails will arrange themselves so that they are facing each other because they are both nonpolar. And up here we have our hydrophilic heads. Down here we have our hydrophilic heads. So this would be like facing the outside. This would be facing the cytoplasm of the cell. Here this would be facing the inside portion of the cell, right? And then we have in here the zone of nonpolar, the zone of hydrophobic. So as stated before, the cell membrane is semi-permeable. So the reason why certain things cannot pass through, if they are nonpolar, they're going to repel back out of this nonpolar area. If things are polar, they're going to be rejected. So that's what makes certain things in here semi-permeable. That's what makes the cell membrane semi-permeable.